everyone, this is Mr. Dempsey. Welcome to video 5-5, Modern Techniques, Part A. In your last video, you looked at how DNA could be used for both storing and transmitting genetic information. Today, we're going to be looking at how the genetic code of instructions can be used to identify individuals and reveal familial or evolutionary relationships. Specifically, we're going to be looking at how DNA fingerprinting works, how gel electrophoresis can be used in this process. And to begin that idea, let's look at a cell. This is a bacterium right here. And that bacterium is covered by these viruses known as bacteriophages. This is actually taken with a special microscope called a transmission electron microscope. And what these viruses lack is their ability to make part of themselves. The reason why is because viruses are made of both protein and DNA or RNA. In order to make protein, the protein coat, you need to have ribosomes, which viruses lack. So all viruses have to infect a host cell in order to make more of themselves. So we see this virus injecting its instructions into this bacterium, and therefore the host cell, the bacterium, will be using its ribosomes to make more of virus. However, over evolutionary time, bacteria evolved defenses against this. And one of the things they've evolved is this ability to recognize that foreign DNA. So you can see that here we have GAA-TTC, that area of the viral DNA. And anywhere a bacterium sees that sequence of nucleotides, that genetic information can be cut. So we first of all, we call those areas of those target areas where the bacterium is going to attack that foreign DNA. We call those restriction sites. And the bacterium has evolved these enzymes in order to cut that DNA, which are called restriction enzymes. So notice the restriction enzyme is cutting that foreign DNA between the G and the A on the top of that sequence of DNA, and then the opposite side as DNA as a double helix, also between the G and the A, going in that anti-parallel direction. So what that would therefore do is it would shred the viral DNA, therefore disabling the virus's ability to infect that cell. Now consider this though. Now that we understand that bacteria have these molecular scissors essentially, maybe we could use that as a technology as a way of cutting DNA. This particular type of restriction enzyme, by the way, is called EcoR1, named after the fact that it's from E. coli, and it's a restriction enzyme, or the first restriction enzyme of this type. So it's restriction enzyme 1. All right. So let's say that we have two individuals. We have individual 1 with a particular allele, which has that GAA-TTC site there, there, and there. And using EcoR1, we could cut the DNA into these 4 fragments, versus individual 2 has a different allele for the same gene, and that person has a different area of restriction sites, only 2 of them. So if we use EcoR1, we're going to be cutting their DNA into 3 fragments. All right, so what could we then do with that DNA? Well, there's a device called gel electrophoresis that will help us in this process, and so you can see a depiction of that. And it's called gel electrophoresis because it has a gel made out of agarose, a type of polysaccharide. And this can be made by students or your teacher. And you can see there's little holes in which the DNA can be put right there. Next, you can also see that there's a red wire over here, which is actually positively charged. And on the back, this is a negative wire. So I'd like you to consider 2 things while you're looking at this picture. First, think about how DNA might have a charge and how we could use that charge to separate the DNA, or to have it move across maybe in this device. In other words, would it move toward the positive or the negative end? Secondly, consider how DNA is broken into these different sizes like we saw on the previous slide. Which pieces of DNA might move faster through the gel? The smaller pieces or the bigger pieces? Think about maybe why that might be. So here's a picture of a gel electrophoresis device that I was running back in 2008 when I was working in the lab. And you can see that I put the DNA into these wells right here, and I turned on the gel electrophoresis device, and you can start to see that the DNA and some other dyes that I used in the process begin to move toward the positive side. Okay, so if you thought earlier about the fact that DNA has a negative charge, that's because of the sugar phosphate backbone. The phosphates of DNA are negatively charged. Also, if you thought about how DNA was broken into those fragments, the smaller fragments will move more quickly through our gel than the larger strands. Larger strands get caught up in that polysaccharide obstacle course and don't move as fast compared to the smaller ones. We can see a depiction of that here where you can see that there's a scale. This is 100 base pairs. So bases such as a nucleotide pairing up to another nucleotide. So we have these pairs. So 100 nucleotides of those base pairs. So that's pretty small. Over here we have 1,000 base pairs, a much longer strand of DNA. And those longer strands don't move as far compared to the shorter ones. So if we look here on this graph, the idea of the migration of the DNA, smaller base pairs will be able to move more quickly through our gel compared to larger ones, which would be up here. Okay, so let's say that we have a crime scene where DNA was collected, and we have two suspects. We're trying to find out who potentially committed a crime. So by comparing the DNA from these two suspects, we're able to find a match. As you can see, crime scene matches suspect 2. And that goes back to the fact that if we have a crime scene that matches a suspect, let's say we have 4 fragments in that particular example, then that could give us a match compared to another individual which would have a different number of fragments. Alright, so now imagine this. You are a forensic scientist and you want to help the police solve a crime. Tiny amounts of blood or semen are collected from a victim. How can you study and analyze it? So to think about that, consider that there are tiny amounts of blood or semen that are collected from that victim. And that may not be enough genetic information in order to run on the gel. It just wouldn't be enough to visualize on our electrophoresis. So therefore, how could we study and analyze that? So one thing you'd want to do is make lots of copies of the DNA. So if you want to copy lots of DNA, how could you do it? What steps would you need to follow, and what chemicals would you use? One of the most common machines that's used in this process is something called a thermal cycler. A thermal cycler, as the name sounds like, uses temperature, specifically heat, as a way to copy our DNA. And also there's a cycling process, which we'll be looking at. So the method that's used with a thermocycler is called polymerase chain reaction, or PCR for short. And PCR is basically artificial DNA replication, where we can take a small amount of DNA and make many copies of it. The way that that process works is, first of all, we need some ingredients. The first ingredient, of course, we would need is our DNA to be copied, the DNA from the crime scene, for example. And that would be shown right here in green. Notice that we're also using a process of heat here. So at 94 to 96 degrees Celsius, that's a high temperature, almost boiling. And you can see in that picture, it looks like the DNA is being unzipped. Normally, DNA helicase would do that, but in this artificial situation, we're simplifying the process and using heat to unzip that DNA. Now, normally, enzymes like DNA polymerase would be denatured by that temperature. However, scientists identified decades ago a type of prokaryote that lives in Yellowstone National Park in hot springs. And that organism has evolved to withstand very high temperatures. And so the organism is called Thermus aquaticus, or as you can see from this abbreviation, TAC for short, for Thermus aquaticus. And that DNA polymerase has the ability to withstand those high temperatures. So as we unzip the DNA, our DNA polymerase, or TAC polymerase specifically, is not denatured in that process. Next, we have primers. Primers are short sequences of DNA or RNA. In this case, we're going to be using DNA. And that DNA primer is going to recognize a target sequence. So you can imagine our DNA primer was AAA. That would recognize TTT here on our target sequence. And that's sort of like if you imagine you wanted to find a paragraph in a book, and you had maybe the first 5 sentences of a paragraph and the last 5, or let's say words, simplified. First 5 words of a paragraph and the last 5 words of a paragraph. And that would be a way to identify a desired sequence, sort of like typing in a Google search. If you put in a few words, you can find actually where that information might come from. Okay. So next thing we need is we need nucleotides. The nucleotides are going to be used for making more DNA, and you can see those are also included here in blue. All right, so we've already done our heating step. That heating step has unzipped our DNA. The next thing we're going to be doing is annealing. Annealing is going to allow the primers to attach to our target. And so, as I sort of alluded to earlier, if you do a Google search by typing in a few words, you're able to find maybe a source from where that book came from. In the same sense, we're using these primers as a way to attach to a specific sequence. The primers are going to be just a few, maybe 10 to 20 nucleotides long, but that might be enough to find our target sequence. Notice that we've also cooled the DNA down to 60 degrees Celsius. That's going to enable those primers to hydrogen bond to the target. Next thing we're going to be doing is adding our Taq polymerase, so the Taq polymerase can begin to add nucleotides to that primer. And the way that's done is we elongate the DNA, enabling the Taq polymerase to add nucleotides on the 3' end, which would be shown here in the blue, and adding nucleotide after nucleotide at 3' end of our primer. The temperature is heated up slightly, so the DNA polymerase, or Taq polymerase here specifically, can add those nucleotides faster. We continue the process repeatedly. That's why we call this a thermal cycler, and that allows the process to happen over and over again until we have hundreds of millions of copies of our DNA, which are now enough to visualize on our gel. So, last point is that we can also use these processes to identify how people are related through ancestry. So DNA is basically a way that we can find those relationships. For example, Ancestry.com and 23andMe are popular kits that people will buy as a way of finding out how they're related to other people. And we can see in a pedigree that might be done in order to find how people share similar traits, but in a more sophisticated way that can be done by comparing the genomes and what type of differences people share among their DNA, similar to that restriction site polymorphism I mentioned earlier. Another way we could do this is compare different species. So imagine you're interested in identifying how a lion, a tiger, a lynx, and a cougar are related to each other. So we could try to use behavior or anatomy or get some other ideas about where they live. But DNA is ultimately the best way to find that out. In the same way, it's also a useful tool to find out how people are related, and that same process can be used to find out how different species are related as well. So, to wrap up, we looked at how DNA fingerprinting works using restriction enzymes to cut DNA. Gel electrophoresis allows us to then run that DNA across a gel and allowing smaller fragments to move faster compared to larger ones. And ultimately, DNA PCR allows us to take small amounts of DNA and to copy it so we can ultimately run that on our gel. So thank you for watching, and I hope you look forward to our next video, which includes more on modern techniques in molecular biology.